So up next is Alex Monell, who is a bioinformatics PhD student at UC San Diego. Alex is jointly advised in the Goldrath and Yo labs, and he also spent time in the Ecker lab as a rotation student, where very impressively he is second author on this beautiful pathogen paper that Travis was just mentioning. So we're excited to hear more about analysis as Alex is going to describe today. Thanks, Alex. Please take it away.
Yeah, thank you for the great introduction, Trevor. Okay, so as Trevor said, I'm Alex. A lot of my PhD research is focused on applying bioinformatics towards analyzing spatial transcriptomics data. So it's very important when we have these biological questions, we have ways to mine our data and answer them. And the purpose of this talk is to kind of point you in some directions towards computational tools that you can use to analyze your own data and show exactly how these computational methods can be applied towards spatial transcriptomics, particularly in plants. So if any of you have started performing spatial transcriptomics in your labs, you might have realized that one of the main bottlenecks is actually just analyzing the data. We have all these commercial providers that are scaling up their ability to provide spatial transcriptomic services, getting millions of cells in an experiment with many genes, and we can get this data super quickly. So we need to have computational pipelines that are robust and able to scale with several replicates or conditions that we're analyzing. And there's two very common types of spatial readouts. We have our imaging-based methods and we have our sequencing-based methods. Our imaging-based methods are usually able to resolve subcellular single-cell resolution, whereas our sequencing-based, some of them actually are single-cell or subcellular resolution, but you might also get these spots and wells. And for the purpose of this talk, we're going to be focusing on analyzing single-cell spatial transcriptomics data from imaging-based methods. Now, as a background to the biological question that we were studying in this project, we were looking at pathogen infection of Arabidopsis and seeing how the plant immune response was used against these pathogens. So we had two types of pathogens that we were looking at. There was the DC3000 group, which is virulent and evades detection from the Arabidopsis immune system. And then there's the ABR-RPT2 pathogen, which is avirulent, and we can see the Arabidopsis leaf mount an immune response against it. So using the VisGen MERScope, which is an imaging-based spatial transcriptomic system, we were able to profile leaves from Arabidopsis from an uninfected mock time point to 24 hours after infection. And so just from these UMaps of the joint embedding between all these spatial experiments colored by their clustering, we can see that in the early time points, we lack this orange cluster that we later gain at the 9 and 24-hour time points. And this is the plant shifting some of its mesophyll cells into an immune active state. And so we'll cover that a bit more later in this talk. Now, as for data processing in spatial transcriptomics, it is quite involved. On the right, I'm showing a hierarchy of the data where we have our entire tissue section. And then if we zoom into a small region of it, we can see all of our individual cells, an image of the nuclear staining or DAPI staining. And then if we zoom in even farther, we can see our individual spots, our individual fluorescent spots. And so when you use a commercial provider for spatial transcriptomics, they'll usually do this RNA spot decoding for you, tell you where all of your spots are, and that's provided to you. So the first challenge that people have to go through is cell segmentation. And to do cell segmentation, that's when we have our image of our cell nuclei and maybe our cell walls, and we can run algorithms to figure out, well, where the cell boundaries are, and then take all of our RNA transcripts that were detected and assign them to cells. So in our study, we have several different time points, and we want to be able to analyze those time points together. And I'm going to show you how to do integration of many spatial replicates or different conditions, as well as imputing observations that you have in single cell data into your spatial data. That includes imputing gene expression for genes that weren't measured in your spatial data. Later in our spatial processing pipeline, we have cell type annotation, figuring out where these spatial neighborhoods of different types of cells are, or spatial landmarks in the tissue, such as the edge of the leaf or the midline of the leaf. And something that a lot of people care about is choosing a very strong target gene panel for a lot of these targeted-based spatial transcriptomics methods. And I'll show how to do that as well. And then as far as downstream analyses go, one very powerful one that everyone will have a reason to implement is spatial differential expression. So when you have your spatial neighborhoods or spatial landmarks, how does gene expression change within different cell types relative to these spatial landmarks? And then we have many other downstream applications like predicting ligand receptor interactions or mapping gene set enrichment or looking at contacts between different cell states. So jumping into gene panel design, everyone has some must-have genes for whatever biological study you have. These could be cell type marker genes. So in our case, we have leaves, there's epidermis, mesophyll, vasculature. We want genes that can help differentiate those different cell types. Furthermore, there's a lot of genes that are important to our biological question, such as stress response genes or immune response genes, or ligand receptor pairing, so we can do analysis on what might be happening there. And so we have these must-have genes, and then we have the entire rest of the transcriptome, or all other genes. And if we have a single-cell RNA-seq data set that is somewhat representative of, or somewhat similar to the same tissue we're going to run spatial on, we can use very clever computational techniques to figure out what set of genes we should add to our must-have genes to have a very strong gene panel that both helps us answer our biological questions, as well as uncovers the heterogeneity that's present in the tissue that we're looking at. So with this tool called Persist, which I've given the GitHub link for on the bottom of the slide, we can train this neural network to take in our must-have genes and mask a large subset of all other genes and pass our single-cell RNA sequencing data set through and try to predict the RNA expression in the single-cell RNA sequencing data set from only a subset of the total amount of genes. So after training this Persist model, what we'll have is a gene panel that contains all of our must-have genes, as well as the most informative subset of all other genes up to our desired panel size. So if you're doing MERFISH and you can only image 500 genes, it will spit out those 500 genes that you should use. Now, going into cell segmentation, this part can cause a lot of trouble for people. Usually, you will not have a super good DAPI stain and cell wall stain or cell membrane stain. You'll usually have a good DAPI stain. And when you have a good DAPI stain, we have object detection algorithms that can segment the DAPI signal. So with this tool called CellPose, if you see the DAPI staining here on the left, here are the predicted CellPose masks based on the DAPI staining. And so if you do have a good cell wall stain, you can also use CellPose for that, and then you know your cell boundaries. That's your best case scenario, but it's not the most likely scenario. So the most likely scenario is when you don't have that good cell wall stain. And we can do our CellPose segmentation on the nuclei, but then there's all these transcripts in the cytoplasm that we're missing if we only segment the nuclei. So we can take our CellPose segmentation and the centroids of all of those cells that were segmented and feed them into a tool called Baser, which can look at all of the transcripts that were detected in our spatial run and assign those transcripts to a cell with a special computational algorithm. And so even if we have a really bad nuclear staining and we can't use our CellPose for nuclear segmentation, we can still put all of our transcripts into Baser, and it can try to predict cell boundaries with a bit lower accuracy. Now, before I get into the next parts of the spatial processing pipeline, I want to just give a an overview of the structure of a spatial transcriptomics data set. And so we have this cell by gene matrix, which is, you'll always see this in your single cell RNA sequencing data. And the only addition to spatial really is your spatial XYZ coordinates at every single point for each cell. And then you might have some cell metadata such as cell type or spatial niche, et cetera. And then we can also have some images that are aligned to our spatial coordinates, like if we have single molecule fish or our cell segmentation mask that we want to include as part of our object. And in our case, we were actually able to label the bacteria that were infecting our leaves and have them in single molecule fish images that were overlaid with our merfish. Now, to integrate several spatial experiments together, we have our mock through 24-hour time points. And we can use a tool called SCBI to integrate these together. And what this does is it removes some of your batch effects between your experiments while trying to preserve the actual underlying biological differences between them. And so it puts all of your cells and maps them into something called a latent space. If any of you are familiar with principal component analysis, that also creates a latent space. And what a latent space is it's a multi-dimensional set of vectors that have a value for every single cell, and cells that have similar characteristics will be grouped together. And in this way, this multi-dimensional vector is a lot smaller than the multi-dimensional cell by gene matrix. So when we calculate neighbors in this latent space, they're a lot more meaningful than if we calculated them on our entire cell by gene matrix. So what this allows us to do is calculate neighbors in the latent space and then run UMAP based on that neighbor calculation and have a joint embedding of all of our spatial cells. We can also do lighting clustering on these neighborhoods since within this joint embedding space. And so this is a very fast tool. You can run it in 5 to 10 minutes. And it allows us, if we wanted to do de novo cell type annotation from our spatial data, which is really neat. And that's a big part of why you may be able to do de novo cell type identification, is if you have that good gene panel that you make with persist from before. Now, if we have a single cell RNA-seq data set and we want to transfer observations from the single cell RNA-seq data set into spatial, we use a very similar tool. It uses the same type of method, and this tool is called GIMVI. And so in our case, we have a single cell RNA-seq data set from our 9 hours post-infection time point, and it's colored by this immune activity score. We want this immune activity score in our spatial data. So what we can do is use GIMVI to facilitate this transfer. And once we ran this GIMVI, made our latent space, calculated neighbors, and done the transfer, we can see this immune activity enrichment in our spatial. We can smooth it over the nearest spatial cells. And this is not only limited to continuous variables like a continuous immune activity score, but we can transfer categorical variables like cell type, for instance. And this also works for imputing your unmeasured counts. So if we had a gene that we didn't measure in spatial because we have a very limited panel size, we can impute what that expression may look like, which is very neat. And this works for imputing RNA counts, attack fragments, or motif enrichment, for instance. Now, I just wanted to highlight how cool it is when spatial becomes multimodal. And so we had this bacterial metagene that allows us to detect bacterial colony positions with single molecule fish and overlay that with our spatial. So at our 24-hour time point with ABR infection and DC3000 infection, we were able to actually capture the positions of these colonies and their size. And what we saw is that in the leaf that was infected with ABR, there was location of these colonies much closer to these hot beds of these spatial niches, or these immune niches spatially. And we can also see that the colonies were much bigger in our DC3000 infection because this pathogen evades detection in the Arabidopsis immune system. Now, this is a very, very important downstream application that I think everyone may want to do with their spatial data. And the tool I like for this is called Seaside. And so spatial differential expression, everyone's going to have a spatial observation based on what biological question you're studying. That's just the whole reason you'd be doing spatial in the first place. So it's going to be your distance to a landmark, your spatial neighborhoods. In our case, we have this immune response enrichment that's smoothed over the nearest spatial cells. And so we can call this an explanatory variable. And what CSI does is it is able to find differential expression along this explanatory variable, whether that variable is continuous or a discrete variable. So we were able to see that within mesophyll cells, ALD1, FMO1, and BON3 were all very upregulated in our immune hot zones. And I think it's based on how you build your computational pipelines, you can make it so that your spatial transcriptomics are a fantastic screening approach to find new candidates for targets to do further validation on. So with BON3, FMO1, and ALD1, we were able to look at them further and see that they, even though they're all expressed in these immune-rich zones, they actually localize into very specific regions. So when we look in our single nucleus RNA-seq data set, BON3 is expressed in this subpopulation of cells where FMO1 and ALD1 are not necessarily expressed there, even though they are enriched in immune-active zones. And we wanted to look closer into these BON3 positive cells and found a transcription factor that's co-expressed with BON3 called GT3A. You can see here in the single nucleus RNA-seq data. And so when we made overexpression lines of GT3A, we saw a decrease in the genes related to the salicylic acid pathway, and overexpressing GT3A led to increased growth of this DC3000 pathogen during infection. So it increased susceptibility to DC3000 attack. And so you have this population of cells that is actually increasing susceptibility to pathogens. And that is something that we're in the process of exploring further as to why exactly the plant would be expressing genes that hurt itself. So I hope I helped inspire some of you on the computational end with tools to use or treating spatial transcriptomics as a screening approach. And I've linked the preprint of this work here that explores some of the things I talked about, plus a lot more. And we also have a GitHub repository for doing some of the things I mentioned here, as well as several other downstream applications that we didn't have time for today. And I'd like to thank Joe, Tatsuya, Hanqing, and Travis for all the help that they provided in this project.
Thanks for this great talk, Alex. This was a really informative, really tour de force of analysis methods for spatial data. So let's open the floor up for questions. If you have them for Alex, go ahead and type them into the chat. So Alex, you mentioned integration of several spatial experiments together. So I was curious, like, are there ways to deal with, morphological differences when you're sampling slightly different sections? And are there ideas about how to put these kind of data on a common, spatial scaffold, an idealized version of the organ, for example, or tissue that you're looking at?
Yeah, that's a great question. So when you have sections in slightly different areas, in my experience, there is usually some sort of transcriptional gradient between the two sections. And so there's going to be slight differences in the transcript expression. But the good thing about SCVI is it will recognize the shared components of certain cell types. And if you have two sets of cells that have a large percentage of genes that they express similarly, but due to their differences and being in slightly different areas, there's this gradient where the gene expression is a bit different. They will still group together in SCVI. However, there will be some difference between them, and you can explore that difference and see that maybe there is some transcriptional difference after running SCVI. And then as far as mapping to a reference, it's always going to be that's always going to be dependent on the quality of your reference. So if you have a reference that is very representative of the samples that you're running spatial on, using these integration methods, you can absolutely map to that. And there are several tools actually for mapping one tissue section to another.
Nice, thank you. So there's an anonymous question that's asking, when you generate the targets, I think this is using this tool that you mentioned to sort of design your gene panel. How many usually give you good signals on imaging-based spatial transcriptomics?
Yeah, so in general, genes that have high predictive power in helping you differentiate different single-cell RNA-seq cell types are going to be ones that are actually expressed, because if they're not expressed, they're not going to have a really high importance score in resolving the heterogeneity present in your single-cell data. So I've only ever seen a couple instances where a gene that I got from Persist or another tool for this purpose gave me something that was just not expressed spatially. And that's usually due to having a single cell RNA-seq data set that is not representative of the spatial sample.
That makes a lot of sense. It looks like there's also a couple questions about differential expression with Seaside. So the first by Sean Robertson asks, can this work with pseudotime, like if that's your explanatory variable?
Yeah, absolutely. So we actually, our immune enrichment score is a pseudotime calculated on mesophyll cells. And with this, we mapped it to our spatial data and then smoothed it over the nearest spatial neighbors so that we could figure out what the immune enrichment was in all cell types, not just mesophyll cells, since we're smoothing over the spatial neighborhoods, and it's like finding regions of the tissue that are immune active. And so you can absolutely do it with pseudotime. That's exactly what we did.
Thanks. And then a follow-up question from Ping Kao is asking, how different are the results in terms of the differentially expressed gene list when you're using particular tools like Seurat or Seaside?
I'm not sure that Seurat has a spatial differential expression tool.
Sorry, I think I misread the question. Actually, they're asking single cell tools such as Seurat versus spatial tools such as Seaside.
Okay.
That makes more sense.
Yeah, so we haven't done, or I haven't ever done a super close comparison of that. I think with single cell RNA-seq, you're always going to be making some assumptions since you can't see the exact tissue slice that you have done your experiment on. And with spatial, I think it gives a little bit more confidence on a lot of tasks, including this one. For instance, ligand receptor interactions. That's something that I think has increased power in spatial because you can see if cells expressing some ligand and receptor are close to each other rather than just inferring it in this like bag of cells approach that you get in single cell RNA-seq.
Absolutely, that's a good point. You have much more confidence that the cell you're seeing is actually that cell, whereas with single cell, you're using the data to annotate what it is. All right. Unless there are other questions, thank you so much, Alex, for this great talk.